You will hear a lecturer in the marine biology department discussing the laboratory protocols for researching bioluminescent bacteria. First, you have some time to look at questions 11 to 17. Now listen carefully and answer questions 11 to 17.
Moving on to the actual laboratory protocol, the extraction of Vibrio fischeri from deep ocean samples is a delicate multi-stage process. In stage 1, our primary goal is purification. Once the seawater is in the lab, we utilize a high-speed centrifuge. The objective here is to filter out any organic debris that might interfere with the clarity of our light readings later on. Once the water's clear, we must ensure the bacteria survive. We do this by carefully regulating the salinity levels in the pressurized tanks. If the salt concentration isn't an exact match for the deep sea environment, the cellular membranes will rupture. In stage 2, we move to cultivation. We place our refined samples into a broth that is heavily fortified with amino acids. This provides the fuel necessary for the colony to multiply. Now, an interesting thing happens here. The bacteria won't glow immediately. We have to wait for a specific population density known as quorum sensing. Only once this threshold is hit do we see the onset of bioluminescent signaling. If the population is too sparse, the lights stay off. Stage 3 is all about the data. We use a luminometer to get our primary readings, but those numbers mean nothing in a vacuum. We immediately upload our findings to a centralized database to see how our strain compares to global benchmarks. However, we cannot trust the machine alone. We also conduct a control experiment using sterile water to verify that the light we are seeing is purely biological and not caused by some stray chemical reaction in the medium. Finally, in stage 4, we reach the dissemination phase. The results are drafted into a formal manuscript, but as with all high-level science, the work is not considered fact until it passes through peer review. This involves a panel of independent microbiologists tearing the methodology apart to ensure it is airtight before it ever sees the light of day in a journal.
That is the end of the lecture. You now have half a minute to check your answers.